At the bottom of your laptop, you move the cursor, you will see a green button, share screen.
Okay, okay. Yeah, I saw that button. Okay, after pressing that?
That you will see your screen, and where the presentation you click, and this will stop.
Oh, yeah, I got it. Okay. Now you can see?
Yes.
It's clear, doctor. You can go, doctor. So nice.
Great. Thank you. Assalamu alaikum wa rahmatullahi wa barakatuh. I'm so happy to be with all of you once again. It's a privilege and it's an honor for me to be talking to you this morning. I'm really thrilled that we are continuing in spite of the pandemic and all that. At least virtually we are meeting each other. It's a real disguise and blessing in disguise. So today I will be talking about my pet topic on biomarkers as valuable tools in clinical diagnostics for breast cancer. I've been working on breast cancer for a long time, but I took this subject in spite of our pandemic, where our Omicron and Delta and COVID-19 is causing too much trouble. And we should be looking at the biomarkers to predict that disease, which is very deadly and devastating the senior citizens of the world. And also now it has come even to the younger generations. But we are not allowed to look at the samples from those patients. So we continue our work in cancer research. And one more thing is that in cancer research, many people have suffered a lot due to this pandemic, because the operations which were scheduled for breast mastectomy or operations for removing cancerous tissues were all postponed, and people suffered a lot. So this was the situation about the cancer. So the topic about biomarkers is how predictive or how diagnostic we can be by looking at the markers. So my topic about this would be how you introduce this biomarkers. The thing is, I'll put it on the full screen. Okay, so we can say what are biomarkers? Biomarkers are specific molecular alterations in the cell DNA, and which can be used as an indicator of a particular disease or some physiological processes and things like that. So these biomarkers can be found in any specimen, such as blood, tissue, body fluids, and biopsy samples and other things. So they can be used as potential tool for monitoring disease and for predicting. Tumor markers, also there are many types of tumor markers, which I'm going to talk about. They can be, it has gone, it's going forward and backward, how to make it. Yeah, got it. So biomarker specimens can be either when the gene is mutated, an SNP can form a biomarker, or amplification of DNA and methylation of DNA. These are the kind of biomarker specimens which we can look at. And especially when it comes to RNA, it is the microRNA or the messenger RNA, which can be predictive biomarkers. Proteins, there are several glycoproteins, CA125, CA50, CA35, and all these are all protein which are glycan biomarkers, which can be overexpressed, underexpressed, and the quantification of these will form, our biomarkers for understanding the predictive or the prognostic value of these. And cells and tissues, like you can take a biopsy sample and look at the DNA in that, or the quantification of the what you call that DNA samples, and Actually, from the cells, we identify, extract the DNA and look at the SNPs. So that's why they are very specific for that tissue or that cancer. So the biopsy biomarkers are great predictors of the status or the stage of cancers. So these the biomarkers can now be classified into diagnostic, prognostic, predictive, or monitoring. Monitoring is when you're treating a person and seeing the chemotherapy is effective or not. You will look at the biomarkers, and when they are lower, then you know that your treatment is going right. Diagnostic is when you know that in the blood circulation, the biomarkers are circulating in large quantities, and you know it is a diagnostic biomarker. And prognostic means when there are small amounts of the protein circulating in the blood, in the circulation of the blood, then you know that the cancer is on, and it'll reach to higher levels, or it can, by your treatment, if you interfere at that level, you can reduce the cancer disease. Predictive is the same as prognostic, which you can predict for a drug response. When you give a drug, whether the level of those biomarkers in your blood has gone down or not. So these are the kinds of 4 types of biomarkers which we can research on, and we have been researching on these. And now coming to breast cancer, you know that breast cancer is very common of all cancers, and it is a leading cause of cancers, deaths in women worldwide. First, it used to be 1 into 8, and now it has gone 1 into 20. So now, even though the cancer deaths have come down because of the advances in therapeutic procedures, still it is a great concern globally, breast cancer. Actually, you know, there are 11.6% rise in the breast cancer cases as diagnosed in 2019, which can increase to 6.6% deaths globally by the GlobeScan statistics. In 2020, there were 27% more new cases, and now it is reported that it is going to be 87,000 deaths, which will be in the future. Even according to WHO, worldwide, 70% of all breast cancer cases will be in the developing countries, like India and other countries. So the treatment options for breast cancer are these. Like surgery is the first option, where you remove the cancerous tissues and give them chemotherapy and radiation therapy. Or if it is typical ER/PR positive breast cancer, then you can give hormonal therapy. And that is also called targeted therapy, where you target those tissues only to destroy the cancer cells in the breast. So we can now see that there is still need to develop biomarkers, because whatever blood biomarkers we have, they have to be validated, and they have to represent the case of the condition or the disease status in the patient. So therefore, there is a great need for sensitive biomarkers to detect in the early stages of neoplastic changes and to detect breast cancer at an early stage. And we thought that, okay, to discover new biomarkers, let us look at methylation status of the DNA. So the promoter methylation panel has listed a lot of these kind of biomarkers. We took some and we started working on promoter methylation biomarkers. So in our studies, alteration of the methyl pattern are on the earliest modification. This is the first modification which occurs when we have cancer developing in the body. So it is, you know, DNA methylation. This gives information which is highly stable and can be easily isolated from the tissue, also from the body fluids. So it's a easy material to handle, to get, and to study. So what is DNA methylation? It's just the addition of a methyl group in the 5 position of the cytosine molecule of the DNA. So methylation imbalance, they may contribute to tumor progression, that is global hypomethylations, or DNA hypermethylation, which can inactivate tumor suppressor genes like P53, P16, BRCA1. If it suppresses, then inactivates the DNA repair genes, and therefore you have a reduction. Whereas when you have hypomethylation observed in neoplastic cells, that is in the cancer cells, they may induce neoplastic transformation. So how it occurs and whether it is hypo or hyper, we need to distinguish between these two. And genomic instability is the really important factor in cancer development. So abnormal chromosomal structures, activating oncogenes, all these are the characteristics of cancer development. Now, the methodology which we used in our study is the biopsy sample collection from breast cancer patients, getting their approvals for ethics committee and from the patient consent, and all that has been done. Formalities completed. Then you look at extract the DNA from the diseased sample, and then adjacent non-diseased sample we take as controls. So we isolate DNA, RNA from those and do the methyl specific PCR and real-time PCR. And we have the results of those where we have the methyl status of a gene called ATM promoter region, where the methylation status represents cancer, breast cancer patients as determined by the methyl specific PCR. And then we do the gel electrophoresis, and it was detected, and the product of methylation are presented in this gel, and unmethylated also was detected by amplification of this 246 base pair product using unmethylation specific primers. And we have 100 base pair molecular weight marker, which was loaded first to identify where the methylation has occurred. So similarly, we did for TP53, which is a promoter methylation status of methyl specific PCR of the breast cancer. So this represents breast cancer patients. Samples were determined by MS-PCR, and then amplification of the product which we found was done on gel electrophoresis of 146 base pair product using methylation specific primers, and unmethylated DNA was detected by the amplification of 247 base pair product using unmethylation specific primers. All the primers, they showed only unmethylated band. So in this case, in the case of P53, it is not a biomarker for breast cancer, whereas ATM gene methylation status is a biomarker for progressive breast cancer. So the difference between the methylation status of the genes will tell us whether it is promoting breast cancer or whether it is not promoting breast cancer. So with this, we can have a predictive and we can have a prognostic or diagnostic breast cancer. Now looking at the clinical usefulness, how useful they can be. Actually, it can determine the risk. You can adjust the risk categorization for the individual. For example, BRCA1, BRCA2 mutation for breast cancer analysis and screening, and prostate-specific antigen PSA for carcinoma of the prostate cancer, differential diagnosis analysis of circulating alpha-fetoprotein or beta human chorionic gonadotrophin, HCG, and also in males with poorly differentiated malignancies of uncertain origin. So these are differential diagnosis and risk determined diagnosis, whereas prognosis, the presence of involved local region lymph nodes associated with subsequent distant recurrence. So these could be prognosis, and prediction could be estrogen receptor, ER, PR, they are endocrine monitoring therapy biomarkers. So there is a need in oncology for clinical care so which can detect the tumor early in the early stages. This is very important for accurate treatment or targeted treatment. It can determine the mechanism of pathophysiology of the stages of the cancer in the body, and it can determine the tumor stage, whether it is in stage 1, stage 2, stage 3, stage 4. And therefore, early detection, benign to malignant tumor progression with the biomarkers, detect residual disease after the therapy, whether disease is still persisting or it has gone completely. So it can detect, the biomarkers can detect residual minimal disease after therapy, which is also very important, because when we think, okay, this treatment has been completed and we are cured of cancer, no, we have to look at the biomarkers for the residual disease. And then we can decide whether to continue chemotherapy and radiotherapy or to stop or to reduce or whatever. So early and sensitive detection, metastatic circulating cells. So this is another very important point for clinical use of biomarkers, because the cancer cells, when they leave the tissue or when they are touched or harmed or something happens, they can, when they overgrow, the cells detach themselves from the main tumor and they start circulating in the blood, and they can go and lodge themselves in any other tissues of the body and form a tumor there. So such thing is called metastasis, and such things should not happen. So we need to look at the cancer cells which are circulating in the blood, and if you find them, then we have to treat those things also, because if it metastasizes, then you have multiple organ failures, or the other tissues, like if it attaches to the bone and grows, then it will be a horrible state for the cancer patients, because they give a lot of pain when they are developing in places where it should not be there. So therefore, understand why people respond differently by looking at the biomarker status. And another thing is, we need to know that the volumes of those biomarkers circulating has to be minimum or maximum or threshold levels. With this, we can decide whether your chemotherapy is working or not. And if it is working, then it has to get reduced from the start of, say, day 1 and day 10 or day 20. Then at the end of the treatment, after 3 months of treatment, then you can check in this time period the biomarker status at 3 times you can check, or 4 times you can check and see if it is reduced. And sometimes what happens is, when they are not reducing, we have a buildup of these biomarkers in the blood samples. That means the patient is not responding to the chemotherapy or radiotherapy. So we start giving higher and higher doses. But the thing is, when you are giving a certain therapeutic, like if you're doing 5-fluorouracil, or if you're giving some other monoclonal antibody treatment, or any kind of drug which you are giving, you start increasing the dose of the drug, which will make the body very toxic. So that is also not good. So to reduce such things, we have to change the therapeutic procedures. If drug A is not working, we need to go to drug B or C. So that is also very important to check with the biomarkers. So biomarkers have a very vast use in clinical prognosis, diagnosis methods to not only to reduce the tumor size or to destroy the tumor, but also to reduce the drug or increase the drug doses. So the main needs will be the need for biomarkers to develop very targeted therapies and individualized treatments. So need for biomarkers for patient selection. That means you have susceptibility factors and resistant factors. a patient can be resistant to one drug or susceptible to one drug. So the biomarkers classify the persons, individuals based on the levels of biomarkers. So these are the main needs and importance of biomarkers in oncological clinical care. So some of the accepted biomarkers which are used as predictive factors for treatment in solid malignancies, you have in the breast the ERPR, BRCA1, BRCA2, I already told you. And for endocrine therapy, where it is triple negative breast cancer, then you have these kind of treatments where trastuzumab, ipanitive, and all these things are used. Whereas for colon cancer, if there is the marker is KRAS mutations, the gene mutations, you can give cetumax and all this panitumab, all these are monoclonal antibody treatments for these kinds of latest treatments for the breast cancer, colon cancer. And for lung cancer, you have the EGFR mutations, which will tell you that tyrosine kinase inhibitors will be very useful to a lung cancer patient when he has or she has EGFR mutations or ALK positive mutations. So these are the kind of information we can draw from the biomarkers and decide on the therapeutic regime of the patients. So in our study, we developed DNA methylation-based biomarkers of reliable gene panel for breast cancer detection. Thank you. I hope I was in time.
Thank you. Thank you, Prof. Kaiser. Thank you. Thank you very much for your valuable talk. Thank you.
Any questions, I'll be happy to answer.
Any questions, please?
From me, is it okay? I already raised hands. Excuse me?
Yeah.
Yes, yes. But
Okay.
introduce
Yes.
yourself, please. Introduce
Okay.
yourself. Yeah.
Thank you very much. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
My name is Rita Maliza. I'm from Andalas University, Indonesia. I would like to say thank you for nice presentation from Ms. Kaiser Jamil. That topic is methylation because in our country, cancer disease around second or third disease in Indonesia. I want to ask about the biomarker use method MSPCR. In this presentation, Ms. Jamil presented there are no ban in the there was no ban in the methylation. That's a conclusion. We conclusion that no methylation. So in the prometer region, and my question is, if we want to do same like Ms. Jamil method MSPCR, which one we focus on, the primer PCR, or we need first we need know sequence in the promoter region or genes sequence gene our biomarker target, and then we design the primer, or something else we need to do for using this MSPCR method? That's my question. Okay.
And that is the latest, when you're looking at DNA and the genes, you have to decide whether you're looking at the primer methylation is the primer region in the gene, right? You have the EGFR gene, then you have the regions, where the DNA which starts the part, which is from 0 to 1, where the codon and this thing, codon 1, codon 2, codon 3, like that, and then the gene part starts. So exactly this is at the primer start region where you do the methyl specific PCR, MS PCR. So here, the one thing is, it is exactly like PCR, but methyl specific PCRs, the region, the start region of that has to be specific. The codons for that region, the nucleotide sequence will vary. You have to design your own primers for that. So there is a software where you can design your primers by using primer designing software. So when you have to do the primer designing software, there is a software which you download it on your computer and you look at that gene, EGFR gene, the primer region, and then you try to design it and the computer will tell you how to do it. So it is a software where you have to learn and use it. So like that, you can design your primers for the methyl specific PCRs. Or if you think that I have done on that, you can use that same gene in another disease and do the same kind of work. So you will get another kind of result. So when it is already published data, you can use that data for another disease for your own, like I have done on breast cancer. You can do on colon cancer, you can do on cervical cancer, you can do on any other kidney cancer, any other cancer, throat cancer, head and neck cancer. So that kind of information can be available in published literature also. Or if you want to do very specific yourself, then you have to learn how to do primer designing and use that primers for MS PCR.
Okay.
Thank you, Prof.
Thank you, Prof.
If you have any more questions, you can email me. My email ID is kj.bmmrc@gmail.com.
Okay. Thank you, Professor. I will contact you later if I have any question and more discussion.
Thank you, Dr. Rita.
Thank you very much.
Make the sound silent, please. Yes, is there any question? If there is any other question?
Yes,
Doctor, yes. Welcome.
This is Avikesh Mahapatra from Odisha. I am a PhD scholar in pharmaceutical sciences. Ma'am, thank you for your nice presentation. It is really quite good information. Regarding actually my doubt is regarding basis pushing like that for testing of these biomarkers. Usually genetic test we are using. So my question is that which type of common genetic test we can use, or whether it is different for all type of disease, cancer diseases like breast, kidney. We are having certain cancer. So the tests are different for others, or we are having some common test we can utilize for the cancer patients.
Actually, the equipment which you are going to use will be the same for all cancers, right? You have a PCR, RT-PCR, and the gel doc, and all the equipments which we are going to use in a system is the same. What is different is when you draw the tissue, you extract the DNA, then that DNA will not be the same for all the cancers, right? There could be some modifications. So you look at a gene. Supposing you're looking at EGFR, then you have to specifically design for EGFR. If you're looking for TP53, that gene, it will have specific primers. Like that ATM gene, you will have specific primers for that gene. So when you do the first routine PCR, the normal PCR, standard PCR, you first amplify that gene. Then you take that product and then go to the MS PCR afterwards. First you get the gene, then you'll have many copies of the gene. Then you do the methyl specific PCR with the new primers. First the primers of the gene, the first step, Second step, when you get that gene product, then you have to have the methyl specific primers for the MS PCR. So there are two steps in this thing. Then you go to the gel electrophoresis, then you do sequencing, and you can do so many other things using that kind of methodology. But the sample and the results will be different for each type of tissue of the cancer. Head and neck cancer will be different, lung cancer will be different, breast cancer will be giving a different result. So whether it is positive result or negative results, you will know when you are doing the gel electrophoresis.
Yes, thank you.
Yeah, it's quite clear now.
Thank you. Thank you, doctor. Thank you, Prof. Thank you, doctor. Okay, next speaker, please. We can call for the next speaker. Thank you, Dr. Kaiser. Thank you very much. If there is any questions, oh, khalas, if there is any questions, okay. If any question, you can put also on the chat box, or you can email Prof. Dr. Kaiser, inshallah, Jamil. Okay. Next speaker, we can call for Dr. Ahmed Mudita from Indonesia, from Adayana University. Dr. Ahmed. Dr. Ahmed. So we can move to the next speaker. Dr. Aman. Dr. Aman Abgandlwar from India. SN